Hi, I'm Mike Yeomans of the Greater Boston chapter of Trout Unlimited. This video is an update on our Embrace a Stream project recorded at our January 2021 chapter meeting. The project is a collaboration between GBTU and the Neponset River Watershed Association that aims to protect and improve the habitat of the native brook trout in the cold water streams within the Neponset River Watershed. In the first half of the presentation, Chris Hirsch details the data collection in 2020 and demonstrates the Embrace a Stream interactive map, an online tool that allows anyone to explore the results of the study. In the second half, Jenny Rogers presents the plans to complete data collection in 2021 and a preview of future restoration efforts. Chris has been on the GBTU board since 2018 and was the environmental scientist for the Neponset River Watershed Association for 5 years. He developed the Embrace a Stream project in 2019. Jenny is the restoration director of the Neponset River Watershed Association. Originally a native of Massachusetts, Jenny recently returned from California, where she attended graduate school at UCLA from 2013 to 2019, and also worked at a water research institute from 2017 through 2020. Thanks for watching.
All right, well, thank you very much, Mike. And yes, as Mike said, I've been around the board for a while now. If you are a regular at the meetings, you're probably sick of hearing me by now. But yeah, tonight we're going to be talking about the results of last year's Embrace a Stream effort and some of the future actions and the next steps that we're about to take with the project. So just a little bit of background on the Embrace a Stream project. So in 2018, GBTU did an evaluation of the different watersheds in our Greater Boston chapter area, and basically looked at what cold water resources existed, and weighed sort of our options of what we want to designate as our home waters, which is really the water bodies that we focus a lot of our restoration work on. And in 2018, GBTU named the Neponset River Watershed its home waters because of all of the cold water resources that are concentrated in the watershed. The Neponset's really unique in the Boston watersheds. Charles, Mystic being the other main Boston watersheds, because it has a fairly high concentration of cold water resources or trout streams. And some of those trout streams, like Trap Hole Brook in particular, are home to very large naturally reproducing populations of brookies. So it's really kind of a special place, and that's why we chose that watershed to be our home waters. And shortly thereafter, we thought it would be a good idea to really evaluate our new home waters and get to know sort of what's going on in the watershed with the trout, and to also understand what sorts of restoration actions are needed to bring back and protect those populations. And so that was sort of the impetus for this Embrace a Stream grant, to really get a baseline evaluation of our new home waters and to figure out a roadmap of future actions. And so at that point, we partnered with the Neponset River Watershed Association. At the time, I was working with the Neponset River Watershed Association as an environmental scientist. So I was working with Bill and Ellen and Gabby. If you guys have been around, you've probably met them, to put together the plan for the Embrace a Stream grant. 2020, we finally launched the grant, or the project, and right off of the bat, we were planning to do stuff spring 2020. Don't really need to explain what happened. COVID-19 basically disrupted a lot of that. And so we were delayed in the starting of the project, unfortunately, but we were able to sort of regroup and figure out ways to do a lot of the project components in a safe, socially distanced way, to collect a lot of the data. So we weren't completely, the year wasn't a complete loss, and we were actually able to get a lot accomplished last year, all things considered. And tonight is really kind of talking about what those results were and what we found in that evaluation. Now we're in 2021, we're starting the second year, so that spring sampling that was planned for last year is going to happen this year. And then we're also launching some other efforts that Jenny's going to talk about really in more detail. And then, as I mentioned before, this whole project came about not as sort of a single one-off thing, but really sort of the base that we're building all of our future partnership actions, all of our future restoration actions off of, really looking at the data to prioritize and identify future restoration needs. Now I've gone too far. So first off, I want to thank all of the volunteers and donors that made this possible. So if any of you donated, we had over 65 donors contribute over $5,000 towards this project, which helped draw in a lot of prize funds and additional funding sources to more than $20,000 to pay for this. And even more valuable than that is the time that people have given to the project. So if I left your name off this list, I'm very sorry. I kind of threw this together last minute, going through some of my past email lists. But yeah, well over 20 people volunteered, got involved, and helped make this project possible. So I just want to say thank you to all of the volunteers, because we literally could not have done this without your efforts. So as I mentioned earlier, the project goals was to really kind of assess the condition of our home water trout streams. We wanted to really understand how the trout were using each stream seasonally, where they are found. We wanted to identify what sorts of problems exist in the watershed, and then develop plans to address each of those problems and prioritize, you know, which problems to tackle first. We evaluated 3 main stressors in the watershed. So the first is habitat fragmentation. Basically, are there barriers in each of these streams that are preventing fish from getting to where they need to go? There's 2 sort of main types of barriers that we think of. One is dams. So dams obviously create sort of like a one-way traffic on a stream. A fish can go downstream over a dam, but they can't really go back upstream. And that causes a lot of issues when it comes time for spawning, or stream sections get too hot and they need to find more optimal habitat. That can create a big issue for them. The other main types of barriers that exist in the watershed are undersized or poorly designed culverts or road crossings. You might have heard of a perched culvert, where the pipe that's carrying the stream under a road, the bottom of the pipe is actually above the stream bed, so it creates like a small waterfall that fish can't get through. Or sometimes if the pipe is too small, it creates artificial increase in stream velocity to the point where fish have a hard time swimming against that current to get up through there. So we went through and evaluated the fragment, or the barriers that exist in the watershed in each of these streams. In addition to that, we monitored water temperature. So as you may know, trout, brook trout in particular, are very sensitive to warming of water. They really need cold, well-oxygenated water. So we monitored water temperature in 30 plus locations throughout the watershed to get a sense of where water conditions and temperature conditions were staying cold enough for trout and what areas were getting too hot. And then the final thing we were looking at is seasonal habitat utilization, which isn't really a stressor, but I threw it in here because it is important to understand sort of where the trout go during stressful times of year. Think peak summer in the middle of a drought like we had last year, versus where they go during less stressful times like in the spring. And the way we did that is through a really cool new technology called environmental DNA, which I'm going to talk a little bit more in a minute. So the dam and culvert assessment was the first component of this project. What we did is we used what's called the NAACC method, the North Atlantic Aquatic Connectivity Collaborative method, to evaluate culverts for fish passage. So some of you are helping out with that project as volunteers, but basically what you do is you measure sort of key parameters about the pipes as well as the streams, and then they have a formula that indicates whether that culvert is going to be a problem for fish passage or not. So we assessed over 38 culverts and over 80 culverts and over 39 dams in each of our cold water streams in the watershed. In addition to that, we monitored temperature. So these little devices you see in the middle here, this is actually an older version of the one that we use, but they're called hobo pendant temperature monitors. And they measure the water temperature and record it every 15 minutes. And so you can really see how the temperature changes throughout the day and then throughout the months and the seasons. And we did that at 34 different locations all throughout the watershed in each of the cold water streams. And we did that April through October. And we used those data to compare what is going on temperature-wise with the habitat requirements for brook trout and brown trout, and really sort of evaluated stream health that way. And the last component of the study, which I thought was really cool and sort of a new technology, is called EDNA, or environmental DNA sampling. This is a new method that's really come out in the past decade, where you can collect a single sample of water from a stream, and you can detect the presence of any fish that you've developed a DNA fingerprint for, or a very specific DNA sequence that's really specific to that type of fish. And it doesn't need to be fish. It could be really any aquatic organism. And people have also applied this to soils and looked at terrestrial animals as well. It's really cool, new, promising technology. But we really utilized it to detect brook trout and brown trout. And so the tests that we were using, the methods have been shown to be sensitive up to 1 kilometer. So what we did was basically break up each of the streams into 1 kilometer sections and collected a water sample at each kilometer and used that to assess whether there are trout present in that stretch upstream of where we collected the sample. And we used a really strict protocol to make sure we weren't cross-contaminating our samples. But it was really kind of fun to get out there and then to collect those samples. So in summary, what we found was the vast majority of the culverts and road crossings in the watershed were somewhat restrictive, which means they may become an issue seasonally if water flows are very high or very low, but are generally passive. Fully crossable, about 17% were fully crossable. So if you think of like a bridge that has that's wider than the stream bed, so it's not having any sort of effect whatsoever. So about 17% of pipes were like that, and just under 10% of the culverts that we measured were actually not crossable or barriers for fish passage. In our temperature study, we found that 4 out of our 8 streams, so half of the streams, contained sections that were still staying cold enough to support brook trout year-round. So the water temperatures never crossed the maximum threshold for brook trout. every stream had at least one section that was cold enough to support brown trout. But it's important to note that a lot of the places where we placed these temperature loggers were placed sort of out of convenience for the access and for volunteers to be able to find them easily and go out to them once a month to download the data. So there may have been deeper pools that exist, or there may have been, you know, sections with a lot of groundwater upwelling that we weren't detecting because of where we placed the temperature monitors. So it's not to say that, you know, this is the definitive condition for the entire stream, but this is kind of a good sense of what's going on temperature-wise in each of these streams. And Jenny's going to talk a little bit more about that in next year's efforts as well. And then with the EDNA, we got some unexpected results. So 15 of the sites that we evaluated were positive for brook trout. So about a quarter of all of the sites that we tested. And none of the sites had brown trout, which was really kind of a strange result that we were not expecting at all. You know, conceptually, most people think of brook trout as being more sensitive to things like temperature than brown trout are. So we thought certainly the areas that are supporting brook trout must also be supporting brown trout, but that's just not the case. And what was also interesting was we thought, oh, maybe there was something wrong with the fingerprint that we were using or the DNA sequence. We partnered with a lab in University of Maine to do the actual genetic work. And we thought, oh, maybe that's the problem. So we actually went and tracked down a fin clipping from a brown trout at a local fishery around here, a local hatchery, excuse me, which we assume it would be the source population for the brown trout that were stocked in the Neponset. And the fingerprint worked out perfectly and identified that fish tissue sample as being brown trout. So it sort of validates the fact that, you know, we did not detect any brown trout. That doesn't mean they're not there. They may be in very small levels or in areas where we weren't able to collect a sample, but really sort of a surprising result to not find brown trout. But the good news was more than half of the streams, so 5 out of the 8 streams, actually had brook trout present. But, you know, this is interesting to see in summary, but it's really more powerful and more interesting to look at it in this map form. So we've created this interactive map where you can see how the different layers and data pieces fit together geographically to really get a better sense of what's going on, and allows you to ask some really interesting questions about how some of these different factors are interacting with each other. So let me see, hopefully this link will work. Are you guys still seeing my screen? Can you see the map? Excellent.
Yes. Yep, can see it.
Awesome. So this is the map. You can find it on NepRWA's website if you just go to neponset.org. Right in the blogs section, right on the front page, you'll see something that says, I believe, trout map or something like that. And you click on that link and it'll take you to this map. And so if you see here, you have each of the cold water streams. So this is the Neponset watershed, this shape here. And each of these are the small watersheds for each of the cold water streams. And you can find the names here. So you have Beaver Brook down here and Trap Hole Brook here, brown in the middle. And you can see the results of the culverts. So these are all the culverts that we evaluated, and they're sort of barrier class.
Hey, Chris, I don't think we can see what you're seeing.
Oh, you don't see, do you see?
We just see the static slide, I think, right now.
Okay, let me get out and try this again. Sorry about that.
No, it's fine.
If it gave you a pop-up, you may have to share the pop-up window.
Yeah, I think that's correct. Yeah.
So,
sorry. So this is the Neponset watershed, this shape here. And then each of these smaller colored shapes are the different cold water streams. And you can find their names down here so you can identify each one that you're looking at. And then you have sort of this list of layers, which is really the data that we collected. So this first one that I turned on is the culverts, color-coded based on if they were a moderate barrier or significant barrier. So you can see sort of how the location of the barrier is really important. So say, you know, down here, to have a culvert that's perched or the significant unpassable culvert means that fish aren't really coming up from the main stem and getting into this whole huge system here, that this system is sort of isolated in a way from that. Versus here, you have a problem, but it's much further up in the watershed. So say these sections are being cut off, but the rest of the stream is accessible to fish that are coming through Willett Pond there. So it lets you, looking at it sort of geographically, lets you sort of contextualize some of the results a little bit. You can throw on, here's the layer of dams that we evaluated. So, you know, while this culvert isn't great, there's a dam right downstream of it. So, you know, until that dam is resolved, that culvert's not really a problem. So when you're thinking about prioritizing actions, you know, you can take those sorts of things into consideration. The dam layer is really cool too, and I'm hoping that we can keep updating it so you can see where we've removed dams. So some of you I know on the call, Gary was there in 2015. This is the dam that GBTU and NepRWA removed in 2015 on Trap Hole Brook. And right after that dam was removed, we actually saw trout passing upstream and downstream through the sites, which was really kind of a cool result. So you can explore sort of where the dams exist in the watershed. On here we have fishery records from Department of Fish and Wildlife. So you can actually see where they've surveyed and found trout. So in 1979, they surveyed there and found 47 brook trout. You can see where we collected our EDNA samples, and you can see where we've monitored temperature. And one of the really interesting things is sort of these next sections. So you can see here the different sections of the streams that had brook trout present. So you can see Trap Hole Brook here. Everywhere we looked, there were brook trout, which isn't surprising at all. But right in this adjacent watershed, Beaver Brook, they were only in this little section right here. You can also turn on the temperature results. So these dark blue sections, these are the coldest water. So this is prime brook trout habitat. The lighter blue is areas that are a little bit warmer than brook trout would prefer, but are still cold enough for brown trout to support brown trout. And then these gray sections are really just getting too hot for any sort of trout at all. But it's really sort of interesting, again, to look at sort of the intersections of these different layers and how they interact with each other, because you have a section like this that, according to our temperature data, is too hot, yet there were brook trout present. Or areas where you get a strong kind of confirmation that brook trout are present in this area that's staying cold, but everywhere else that's too hot, there's no trout at all. So really sort of cool to dive into these layers and dive into the results. And that's really sort of what Jenny's about to talk about, is what the next steps are and making sense of all of this and sort of prioritizing next actions. And so I encourage everyone to explore this website on their own. If you go to neponset.org, my internet will let me.
Mike also put the link in the chat.
Oh, excellent. Yeah, it's right here if you want to find it. Native Trout Restoration Map. It's right here on the front page of neponset.org. So with that, I'm going to turn things over to Jenny. I'll stop sharing my screen, and she can fill you in on sort of what's the next steps and how do you make sense of all of this.
Super. Can everybody hear me?
Yes.
Okay, awesome. And can everybody see a slide that has a snowy brook in it and says what's next?
Yes.
Okay, in the right presentation mode?
Yes, indeed.
Super, great. Okay, so thanks, Chris. That was an awesome overview of the genesis of the project. I didn't know some of that information, so it was great to hear you go through it. So hi, everyone. I don't think I've met most of you. I'm new to NepRWA. I started just before the holidays. And actually, as they said when they were giving my bio, I've done most of my professional career out in California. And so I'll use that as my get out of jail free card if I mispronounce any of the brook names or say something silly. So Chris, feel free to jump in if needed. So anyways, Chris just talked about the origin of the project and the awesome data that was collected last year. Really impressive amount of data actually to be collected in just one sampling season. So I'm going to talk about what's next. So we're going to do some more sampling in the springtime. So I want to go over some of the plans that we have, some ways that we might analyze the data, and then some things that might go into the final report, which is due in September of 2021. So just under a year left for this project. And this was actually the first place that I went to when I moved here and started working at NepRWA, Trout Brook right after that snowstorm. And I just thought, wow, this is really beautiful. So anyways, I'm glad to be a part of this project. Okay, so springtime sampling, and it's coming up faster than we imagined. So first, before I get into these bullet points, what I realized based on Chris's talk when he was talking about seasonality is, I didn't even put that on here, but we definitely want to resample some of the EDNA in the sites that they did over the summer, because we are interested in if the fish populations are typically staying in the same areas throughout the year, or if they are showing seasonal trends and maybe moving certain places during the stressful summertime temperatures, and then taking advantage of the cooler spring temperatures and using different parts of the watershed. But what I really wanted to focus on for this talk were how we plan to kind of further investigate habitat suitability at sites with quote unquote curious findings, and then how we want to continue to document trout distribution within the Neponset River watershed. So what do I mean by curious findings? So as I'm sure most of you know better than me, trout, like brook trout, like most trout, prefer cold temperatures, right? They're cold water species. And so a curious finding might be an area that had very high temperatures over the summer, and yet there was a trout population present. And then similarly, a curious finding might be that there were cool summertime temperatures, right? So it might be considered a refuge habitat, and yet there were no trout populations present. So those are two places that we want to do some further investigation in this springtime. And then in addition to that, we want to continue to document the trout distribution. So Chris mentioned that the focus of the summer sampling was really on the cold water streams. But he also mentioned that the main stem can be used as a corridor, right, for trout to get from one tributary to another, or one cold water stream to another. And so we're curious if they are using the main stem, right, and if they are traveling between tributaries. So we might want to add some sites in on the main stem. And then finally, some sites on the east branch of the Neponset River, or tributaries in that area, where we really only looked at one stream in the east branch over the summer. So getting into this, so first, some of these curious findings. So what are our plans here? So areas that have high temperatures based on our monitoring, and yet there was trout found based on the EDNA analysis. And when I say trout, I'm referring to brook trout, because as Chris mentioned, we actually didn't find any EDNA for brown trout. Doesn't mean they're not there, it just means we didn't find them. So in general, I'm referring to brook trout. Okay, curious finding number one, high temperatures and trout present. So the main question that we have in these areas is, given that we did monitor some pretty high temperatures, we want to know if the fish are able to find refuge habitat in these areas. And when I say refuge habitat, I mean cold water areas, or if they're just stuck in these warm temperatures and they're stressed out through the duration of the summer. So the example that I'm giving here is Ponkapoag Brook. This was one of the sites right next to the confluence with the main stem. And if I direct your attention to the graph at the bottom of the page, this is just the raw temperature time series that they monitored over the summer. So we have temperature in degrees Celsius on the y-axis, and we have time on the x-axis, and then we have all of our temperature results in the lines. And then I put a big red horizontal line at 20 degrees Celsius because that's about the temperature that starts becoming stressful for brook trout. So any temperatures above 20 degrees Celsius are going to start getting a little stressful. And you can see some of the statistics up top. The maximum temperatures were even approaching 26 degrees Celsius. So super stressful for these species. And impressively, the time spent above 20 degrees Celsius was 431 hours. So about half of this time that we monitored temperature, these fish were experiencing stressful conditions. And so we want to know, well, then why did we find such a strong signal of trout in this area? And we want to know, given that there are so many trout here, were they able to find refuge? And they could find refuge if there's two things, right? If there's lateral connectivity and longitudinal connectivity. So I just wanted to define those two terms before I move on. So lateral connectivity, and let me just say, you guys would not believe how long I spent of my Saturday making this slide and trying to build a willow tree out of the various icons and shapes on PowerPoint. Lots of my Saturday went to this. So anyways, I appreciate my willow and my cattail marsh. So anyways, lateral connectivity means that the main channel is connected to the floodplain, is connected to the side waters and the back waters and the edge waters. So for example, if the fish are hanging out here and summer rolls in and it starts heating up, they're able to move from that stressful area to a lateral position in the stream. On the contrary, longitudinal connectivity is a little different. By the time I got to this slide, I was tired of PowerPoint, so it's a lot simpler. I didn't make it as beautiful, but longitudinal connectivity, as opposed to referring to the cross-section of the stream, actually refers to the connection headwaters to estuary. So can the fish move up and downstream? So again, if the fish are hanging out here and it starts heating up, are they able to move downstream? And in general, they would be, unless of course, like Chris mentioned, there's a barrier. So if there's a dam or a culvert, or a natural barrier also. So, you know, a steep step pool or waterfall or whatever. So anyways, back to this slide. So what we want to do in the springtime is look at the lateral connectivity and the longitudinal connectivity and do some additional analysis. So we want to see laterally across the stream, are there different habitats that have different temperature variations? So even if where the temperature logger was in a particularly warm area, maybe nearby, like Chris was mentioning, there's a lot of cold water upwelling or a very shaded area that could be a refuge. And then maybe they're able to move longitudinally. And so we'd want to do a microhabitat analysis above and below significant barriers to see if the fish are able to find refuge elsewhere. Okay, so that was our habitat suitability at sites with curious findings for the high temperatures. So now we are curious about sites where there were cool temperatures and yet no trout population present. So in this area, same sorts of figures on the slide, a different site though, so the data is a little bit different. So now you can see with our temperature time series that throughout the duration of the temperature monitoring, for the most part, it stayed below 20 degrees Celsius. Okay, so in general, the temperature was not passing over that thermally stressful threshold. Maximum temperatures were just about 22 degrees Celsius compared to the almost 26 degrees Celsius at that last site. And most impressively, the time above this threshold of 20 degrees Celsius was only 19 hours. And that's throughout the entire summer. Whereas at that previous site, remember, we had over 400 hours of stressful temperatures just in the month and a half where they were doing the monitoring. So in general, this looks like it would be a thermally suitable site for brook trout. And yet, based on the EDNA analysis, we didn't find any. Again, it doesn't mean that they're not there, but we didn't sense any. And so what we might want to do here is a barrier analysis from the nearest population. So we could use the dam and culvert surveys that were done and try and figure out maybe the issue is brook trout just aren't able to repopulate this area. But we'd also want to look at other stressors, right, and try to figure out why they aren't here. So if it's not a barrier, and if it's not a temperature issue, possibly it's a habitat issue. So maybe there's not, gravelly riffles that they need to spawn or deep pools. It could also be a hydrologic issue, right? So maybe there's not enough water at the right times, or there's too much water at the wrong times. So different things that we want to look into. It could be water quality, and it could be biotic stressors, right? So there could be, maybe there is not a lot of cover and there's aerial predation, competition, etc. So these are other things that we want to look into this springtime. And one interesting thing to kind of keep in the back of our mind at these sites that do have cool temperatures throughout the summer, for whatever reason, like it could be lots of groundwater upwelling or lots of shading or whatever, there are potential sites for stream translocation in the future. So if we think ahead, thinking about climate change and some of these other sites that are getting very hot, if they continue to get hotter, they might become too hot for the brook trout, right? And then we might want to move some populations, and this could be a refuge area that we kind of keep in our back pockets as a place where we could seed a population. Those are our curious findings. So next, what we want to do this springtime is continue to document trout distributions. So we talked a little bit about eDNA sampling on the main stem, and then doing some eDNA sampling on the east branch of the Neponset River or some of the tributaries. So starting with the main stem. So what you're looking at here, you guys all probably know this much better than me, is an outline of the Neponset River Watershed. All the blue lines are the streams, and anywhere you see a dot is where they did eDNA sampling over the summer. Okay, so a black dot, the small black dots mean no signals found. Again, this doesn't mean that the trout aren't there. It just means that we didn't capture any eDNA in our samples. And the bigger and the redder the dots get, the stronger the signal, which essentially means either the population is larger, or the fish were closer, or the fish are bigger. For whatever reason, there was a lot of DNA in their sample, and they were able to sense that when they did their eDNA assays up at UMaine. Okay, and so I want to zoom into this dot in the middle. Chris, I think he talked about this one, but this is a strong signal at Ponkapoag Brook. Interestingly, this was actually the first example I used as the site with high temperatures and yet a large population found. And it's kind of an interesting site because it's right at the confluence. And so it first makes us wonder, if the trout are so close to the confluence, are they using the main stem as a corridor to get to other areas, particularly because based on the eDNA signals moving up the stream, except for this one site here, we didn't signal moving up the stream. And so we might wonder, are these fish also using the main stem? And this would be a means, I think I mentioned this earlier, as a way for trout to get from one area to another. So for example, this trout population on Ponkapoag Brook could migrate up and get to another cold water tributary elsewhere further up the stream. So we want to do some sampling along the main stem to see if trout are able to use it as a corridor. And just because they're on the main stem doesn't necessarily mean the main stem has to support all of their life history phases. Like they don't necessarily have to spawn in the main stem, just a question of if they're able to use it for migration. Okay, so and next, same map, but I've moved my little box to be lower in the watershed. So we're curious about doing some additional sampling on the east branch of the Neponset River. So over the summer, we looked just at this one tributary, Beaver Brook. And you can see a lot of black dots, which means we didn't find much of an eDNA signal going up until the very tippity top of the watershed where we found a weak signal. But just because it's a weak signal, it just means that someone's there, right? So there's brook trout present. And so it kind of makes us wonder, what about the rest of the tributaries? So I'm going to zoom in on that site. So this is still Beaver Brook, right? We still have our black dots. As we go up, we didn't find any EDNA signals, and here's our weak signal at the very tippity top of the tributary. And now I've added in where we did the temperature monitoring. And so interestingly, temperature monitoring was done right next to the location where that eDNA sample was taken. And so this corresponds to the top graph where we have our same temperature time series with our same 20-degree threshold. And you can see at this site, temperatures stay well below 20 degrees Celsius. And so it makes sense that the fish might be there because it was a thermally suitable area. But interestingly, as you go down the stream and we didn't see any eDNA signals, and we look at the other three temperature monitoring results, we see the temperatures far surpass the 20-degree threshold. In fact, they even approach 30 degrees Celsius, which is much too hot for brook trout. And so we might wonder, even though some of the other tributaries in this might not be designated as cold water streams or trout streams, they might have some similar patterns where there's a few areas that are cold enough to support brook trout, and we want to do some further sampling in these areas to determine if in fact there are some populations elsewhere.
Hey, I have a quick question.
Mhm.
It's Andre. Just Chris or Jennifer, can you remind us when the DNA samples were taken, just in relative to those temperature graphs?
I think they were in July, right, Chris?
August.
August, right?
Yeah.
Okay. So just the reason I asked is it seems like there may have been there could have been a mortality event associated with those temperatures, right? Around the time that we took those samples. Just curious, just if that is true.
Yeah,
Might be true. Yeah.
that's definitely possible. So eDNA, it's a great technology for surveying, but it's not perfect. And so if you get your timing off, you know, if the fish moved on or they died off, you know, earlier, then you wouldn't pick up the signal. Similarly, if they're too far away from where you sampled, then you wouldn't pick up the signal. So definitely a negative eDNA result does not mean that the fish aren't there. It doesn't mean that the fish were never there. All it really means is that they didn't pick up the eDNA signal in the sample that you sent them in the lab. So definitely, there could be all kinds of reasons. Good point. Okay, so what's next? So what I just went over is what's next for sampling in the springtime. And then of course we have to put together some ideas for restoration projects. So restoration projects, some of the obvious ones include barrier removals like Chris mentioned, shading, riparian area restoration, essentially making habitat more suitable and increasing the connectivity. We also might want to put together future studies like I talked about earlier. So if it's not a barrier issue, and if it's not a water temperature issue, could it be water quality? In which case we might want to start thinking about stormwater. Could biotic interactions be the issue? Invasive predators or predation? And then is it hydrologic impairment? So is there enough water at the right time? And of course, this last one is going to be a bigger and bigger question as our precipitation regimes continue to change over the years. So some great embrace the stream outcomes that are really exciting. Of course, this means GBTU members and NepRWA staff get to have lots more opportunities to collaborate on fun projects in the future, because essentially we want to put together a laundry list of things that we can do over the years. There might be some great change if brook trout distributions lead to additional stream protections. And then finally, what we all really care about is the ecological implications if we reduce stressors to enhance the resiliency of our native trout toward the changing climate. And then the last bullet I wanted to mention is that what's so great about trout, not just brook trout, but all trout, is they really use all of the stream throughout their life history phases. And so if you improve stream health for trout, you're really improving stream health for all in-stream communities. So all the little critters win. So definitely get involved. We're going to need a lot of help this springtime. I know with COVID it's a little hard, but outdoors, everyone wears masks. You saw from some of the pictures last year. So some of the tasks that we'll be doing this springtime are the same as last year. So deploying temperature loggers, collecting samples for EDNA testing. And then we'll be doing some different kinds of field work, like some surveys of hydraulic habitat and some surveys of stream temperature. Thank you once again. I know Chris showed this slide earlier to all the volunteers who helped last year. This was all before I got here, so everyone really did much more of this project than I did. I'm just here talking about it. Thanks to the people at University of Maine for their EDNA analysis, and then of course all the donors. And that is all that I have. So here's our contact information, and I think I'm gonna pass the posting back to someone. And definitely type in questions or ask any questions that you guys have. Plenty of time.
Yeah, Jennifer and Chris, we will, when you give us sort of the heads up on volunteer dates and opportunities, we'll make sure we publish that on the GBTU social media. And I'm sure, Nancy, I see you're on board. You'll probably do the same with NepRWA also. So stay tuned for timing for some of those volunteer opportunities. And by the way, really great presentation by both of you, really thorough. It's great to see where the money and the volunteer hours went, and the curious finds will be some of the detective work that you have to do.
Yeah, let me echo what Rui said. Chris and Jennifer, that was just an outstanding presentation. And for those of us who were there in the beginning, this is so gratifying to see that this Embrace the Stream has yielded such, superb results. And I think a lot of us are looking forward to when we can get out again as a group and join you in doing some of the field work. I did have a question though. On the stream where you showed the presence of brook trout only at the headwater section, is when you mentioned the timing of it was August, I could imagine that if you did the eDNA analysis in, let's say, May, when those downstream segments of the stream would be colder, you might see that it's populated throughout. And then as it gets warmer, those trout are pretty good at moving upstream until they find the suitable, temperature habitat. So I was just thinking a really nice thing to do this year would be to make sure you do the eDNA analysis in a different, time domain. Do it early in the spring, or maybe even again later in the fall when the temperature goes back to a much cooler level. But boy, it just prompts so many questions and opportunities. So thank you again. I'm so glad I got a chance to see this.
Yeah, definitely. So like Chris mentioned, they did the eDNA sampling in August. So the idea was, let's see what the distribution looks like during very stressful times. So the next time that we're planning to go out is the early springtime. So late March, April, maybe early May, and see how the seasonality affects the distribution of the brook trout.
Yeah, it was a pretty brutal August. If we remember, it was pretty dry.
Yes, very dry.
We tried to take some samples out of mud instead of water. So yeah, it was tough.
Yes, I'd just like to add to what was said. I thought it was a great presentation, both Chris and Jenny, and my daughter and I were sort of super involved in doing data collection, the temperature monitoring, and the eDNA sampling, and it was actually great to just get out of the house occasionally. There was, I think, the second time we went out to collect the data temperature data. My daughter said it was the first time in a month that she'd been out of the house in the car because of COVID. But also interested that you showed the data from Tubberack Brook, which is actually where we collected the temperature data, and that I was sort of hoping that we would detect brook trout there, because it just did stay amazingly cold the whole summer, but also maybe not super surprised because it was such a piece of skinny water. We went after we realized that further downstream was getting getting very warm, but that little point on Tubrek Brook was staying cold. We went sort of to see if we could scout out where the source of the water was, and where the cold water was coming from. And so, you know, initially we said, well, let's go to the source, because it looked like it was flowing out of a little pond. And we found the pond, and then we sort of tried to find where the water came out, and eventually found the stream further down with a culvert, and it was completely dry, and, you know, there was no water. And then we went further downstream, and then there was no water. And eventually we sort of narrowed down. We found one road crossing where there was water just upstream from where the temperature monitor was. But the distance from there to the next temperature monitor, you know, downstream where it was warm water was, you know, only about a mile or so. And that actually goes right through a big wetland, where I think the water gets very warm. There's a place called the Fort Factory Brook Reservation, which we didn't actually go and scout out, but I assume there's a barrier there because it's the site of an old mill. So there's almost certainly a barrier there. And I don't know how difficult it is to remove barriers when they're, you know, historic significance like that. Presumably that's very difficult to remove a barrier like that. It's great to see how it's all connected together, and how the culverts and the lack of water and the EDNA sampling, it all ties together. So it's really good. We had a lot of fun participating too.
Yeah, I was really happy to have you guys. You were one of the more reliable, kind of excited volunteers, so that was great. Thank you so much for helping. And yeah, I think looking at flow would be a really interesting parameter as well, because that system in general, Mill Mine, Brook, Tubrek, has a lot of municipal water withdrawals from the local towns. And so it's not uncommon for parts of that brook, like you said, to go completely dry, especially in the summer as people are, you know, watering their lawns and golf courses are in operation. And, you know, there's so many different demands for water on the landscape.
Right, where that temperature monitor was on Tubrek Brook, the flow is remarkably constant. But each month we went back in the summer, we were always kind of wondering, is there going to be water flowing? And you know, amazingly, it was pretty much the same every week. So it's coming from somewhere, you know, some spring somewhere, I think.
Hey, Chris, I have another question for you. Given all of this fine work, have you considered engaging the state of Massachusetts to do some electroshocking in those brooks where you have found trout, to get some sizing information and population density?
Yes, so we've been working with Adam Kaltza with the state, and he's agreed. We were more interested in some of the smaller tributaries that don't have current cold water designation, to basically have them formally document that, the presence. But we didn't talk specifically about having them document size or population density, but that would be an interesting follow-up, and I bet they would be interested in that as well. We've been sharing the temperature data with them and sharing the EDNA data with them, and yeah, they're very interested in this project.
Great.
I'm gonna check the chat if there were any questions in there.
I didn't see any. There's a few couple of comments.
It's a pretty small group, and no one seems to be too shy, but if you do have any questions, I don't know, does anyone from NepRWA, other than Jennifer, have any comments that you want to make from your organization's point of view? Go ahead, Rory. Just pull off mute.
Yep, I was going to say that the Beaver Brook location that you were looking at, the part that has the, I guess, presence of trout, it's really the headwater of Beaver Brook, and it comes right off of Moose Hill, which is the Moose Hill Sanctuary in Sharon. And the rest of Beaver Brook, where we didn't see the trout, it runs right along the train track, the I-95, I'm sorry, the Providence to Boston main rail line. And that section of Beaver Brook has very little foliage covering it. It's very exposed to the sunshine, and I can see why that would be a location where trout would not be too happy to live, certainly in the summertime, perhaps in the earlier part of the year. And it is interesting, the temperature graph showed very clearly what happens to that section of Beaver Brook in the summer months. So I'm not surprised, but it is interesting that that very short section of the headwater would actually have trout in it. And how did they get there? I guess they must have gotten there a long time ago. And Declan was with me when we surveyed that whole section of Beaver Brook, and we could see very clearly that that was a lush area to be a trout. But once you get through that culvert and start heading downstream, it's not a place I would want to be. It's just too hot.
Yeah, that area up there is fascinating. Just the landscape is so strange. There's these rolling hills and then pools of that are just freshwater springs, essentially. It's a really cool area. I recommend anybody who's interested in sort of geology and springs to go check that out, because it's wild. But I don't go into the cedar swamp. Declan can attest that we collected a couple of the more remote samples that we thought that volunteers wouldn't be interested in getting, and that was probably the most intense hiking I've ever done in my life. It was insane. And I lived in Costa Rica for a year chasing monkeys, so I know jungles, and this was beyond anything I've experienced.
Hey, Chris, Tom Palmer has a comment that he said state shocking happened in Pine Tree Brook a few years ago. So did that just show, did that show any of the brown trout, or do you remember what the results were?
I think it was just brook trout, but don't quote me on that.
Okay.
Yeah, I think that was in 2015. They went and shocked, because I guess we knew there were brook trout there, but the state was never able to find them. And as I understand it, this was before I was at NepRWA, but from what I understand, we basically walked them to the spot and said, shock right here, this is where the trout are. And lo and behold, they found a whole bunch, all age classes too, which is really cool. So that's a sign that they're reproducing there.
Very nice.
They still stock Pine Tree Brook. What is that doing to the? They do still stock Pine Tree Brook. They stock it at the location right at the flood control dam. I'm not sure what impact that's having on the native population.
Do you know what they stock it with? Is it just browns, or do they stock it with brook trout too?
Brook trout, as far as I'm aware.
Yeah.
I've caught brook trout in that like floodplain area. I've caught stock brook trout there, but right before that, I've seen native brook trout. So it was just interesting.
Yeah, that's a whole can of worms that I don't know if we want to dive into right now. But yeah, I don't know if there's any sort of impacts. And yes, Tom did photograph a brown trout on the main stem, which I was hoping we were going to find brown trout, a signal of them this time around. Because if you look at the temperatures on Pine Tree Brook, pretty much everything except Pope's Pond is cool enough to support brown trout. And it's sort of a dream of mine, if you could remove the Baker Dam and you could open up Pope's Pond, you could essentially create a condition where you could have sea-run brook trout out of the Neponset, which would be insane. There's a lot of what have to happens in that, but
Oh yeah,
we can dream.
you got Steve excited. So we're a little bit past 8 o'clock. I don't know, any closing comments or thoughts, or otherwise, fantastic presentation. Looking forward to the volunteer opportunities for NepRWA and GBTU to work side by side in the stream again. And Chris, congrats on your new role and continuing to support this Embrace the Stream project. And Jennifer, welcome back to the East Coast, and and joining NepRWA and all the work that you're going to do with us over the years.